Okay, welcome to Marine Ecology. This is the first of two videos. I like to break it apart and chunk it into manageable sizes. This one's going to focus on individuals and physiology, populations, and then part of community. Then the rest we'll cover in slide presentation part two. Ecologists tend to think of systems in terms of this ecological hierarchy. So there's many different ways you could look at a kelp forest ecosystem. You could look at an individual, a red alga, a Garibaldi fish, an abalone, a sea otter. Or you could look at the population, all of the sea otters living in this particular location that could potentially interbreed with each other. The community, all of the different species that live in that area that interact with each other, brittle stars, rockfish, giant kelp, etc. The ecosystem looks at the interaction between the living and the non-living things, so light availability, nutrient uptake, things like that. Biomes then are a collection of similar types of ecosystems. So not just the coastal California kelp forest ecosystem, but combining it with ones that you would see in Alaska or off the coast of Peru or something like that. Similar habitat types throughout the world. And it could be even more broadly defined like coastal ecosystems. Adding all the biomes of the world then gives us the biosphere. So from the individual perspective, we're going to focus on physiology, so physical adaptations to the marine environment. We look at this in terms of homeostasis, maintaining consistent internal conditions, even though the outside conditions are variable. We look at in terms of tolerance limits. What can that organism tolerate? How salty or how low of salinity can that tolerate? How warm, how cool? And all of these things feed together in the concept of the niche. So in organisms adapting to the marine environment, they have to think of things like how viscous is the water, how buoyant are they? So in order to get through the water, a streamlined body helps. So you can see that clearly on this tuna, the Ferrari of the fish world. And they have fins for maneuverability. You can see the pectoral fins are off to the side, and that helps them maneuver themselves around in search of prey. The finlets along the tail end tend to reduce drag. So as the water goes beyond the bulge of the body, this helps to reduce eddy turbulence on the backside of the fish. And then more of the fish's propulsion is generating thrust. It also helps to have a high aspect ratio, a very, very tall and skinny tail. So you can see the one that's circled here. These are the tunas, very, very tall, very, very skinny, compared to things like mackerels and things like that, where it still can go quickly, but not quite as quickly because there's more drag. So by having a tall, skinny tail and a structure called a caudal peduncle that is very, very stiff as the fish wiggles its tail back and forth, more of that then generates moving the water backwards, which then propels the fish forward. They also possess a structure called a swim bladder, which helps them maintain buoyancy in the water column. So without this, the fish would sink because bones and scales are heavier than the water surrounding them, so they would sink downward or have to be perpetually swimming upward. But by having a swim bladder inside, they can extract gases from their bloodstream and then move them into this bladder and then use muscles to expand or contract that. And then that bladder, that balloon basically inside of them, can help to neutralize their buoyancy and even have them move up or down in the water column without having to swim. For small microorganisms, this takes a different shape. So we've mentioned that diatoms, which you see on the left here, have these glass houses or frustules made of silica, and the silica is heavier than water, so they would tend to sink. However, these oil droplets that occur within them kind of counteract that negative buoyancy and helps to hold them up in the water column so they can stay up near where the sunlight is and photosynthesize. For zooplankton, they have these feathery projections that they can extend outward, and it kind of slows their descent. And then when they want to move up in the water column, they'll take the antennae, which you see at the top of the slide, and they'll kind of flex them downward. You may have seen that in labs if you found some copepods in your water sample, and that helps to propel them upward. So homeostasis is a concept of maintaining constant internal conditions, even though the external environment fluctuates. So if a fish swam up and down through a halocline, that would be a fluctuating condition. It might get more salty or less salty, and it has to be able to adapt to that. And this can be for a variety of things: temperature changes, water, if you're a terrestrial organism, ionic concentrations, and it involves something called negative feedback around a set point. And that's basically, it opposes the environmental change. So if you have a thermostat in your room and it gets too hot, the air conditioner will kick on. That's negative feedback because the temperature is too high, so it cools the temperature. Okay, and if it gets too cold, then the heater will kick on and warms it up. In the human body, our set point is 37 degrees Celsius. And our hypothalamus in the brain senses that body temperature and signals our heater to kick on. If it gets too cold, you increase your metabolism or you start shivering, and then that warms you up. So heat's produced, the body temperature rises. If you reach the set point, it kicks off. And if not, it continues to oppose that change, the cooling of the body, to warm it up. But so temperature is the easiest to explain, but it's not just temperature. So here's another example. In fish, some of them are going to move between freshwater and saltwater during their life cycle. So this is an anadromous life cycle. Salmon swim up into freshwater, lay their eggs in freshwater. The very, very young ones have to be able to deal, the larvae have to be able to deal with freshwater conditions. And then they go downstream until they hit the estuary. And then they have to be able to adjust their salinity regulation to be able to deal with more and more and more salty conditions until they're completely marine. The Pacific salmon spends 5 years or so in the ocean, deals with fully saline conditions, 35 parts per thousand of salt in seawater. But then it reaches a point where it knows it's time to reproduce, swims up through the estuary, adjusts its salt regulation, moves into freshwater, adjusts its salt regulation, swims all the way up to its natal stream, um, reproduces, lays sperm and egg. The sperm fertilizes the egg, and then the fish dies. And the fish actually fertilizes the water, that then there will be higher nutrient content for the larva moving downstream. So more algae, more bugs and things like that for them to eat. So the homeostasis part of this is being able to adjust your salinity, even though it's going to vary during your lifetime. And the way that they do this is marine fish must drink in order to acquire water. And then they lose water through the gills and through their urine. In contrast, freshwater are living in water that's less salty than they are. So they have to take up water through their gills, they lose it through their urine. This is because the salt concentration in the cells is just less than 1%. So a marine fish living in 3.5% salt, it has to actively take up the salt and get rid of the sodium and the chlorine. You see that on the bottom left picture. It accidentally takes up the salts from seawater, and then it has to get rid of the extras because the marine water is more salty. 97% water. In freshwater, it's 0% salt, 100% water. So they're constantly receiving extra water and they have to get rid of as much of it as they can and they have to actively take up the salt. So these two different regulation mechanisms are basically the opposite, but something like the Pacific salmon can change from freshwater to estuarine to then marine, and then later in its life from marine to estuarine to freshwater. Temperature can be something that organisms deal with. One of the extreme examples is that Antarctic cod can survive in water that's below 0 degrees Celsius because there's so many salts dissolved in there, the water can actually get to about 2 degrees below 2 degrees Celsius below zero before it actually freezes. So they have to possess a glycoprotein in their blood that acts like an antifreeze and keeps their blood from freezing because the water is so cold. For mangroves, they basically are taking up seawater as their water source, and then they, like all plants do, will take up water, but they have to get rid of the extra salt. So they'll excrete it. Some types of mangroves will excrete it through their leaves, as you can see on the right-hand side here. And that helps them maintain water and salt balance to maintain homeostasis. For oxygen, the water tends to not have very much oxygen in it. And so fish have to be extra efficient at taking up water. So as you can see on kind of the top left, the diagram of the fish, the water comes in through the fish's mouth and then it goes out both sides across the gills and out the back of the operculum. This little flap covers it and then the water goes across the gills. Now, as the water goes across the gills are basically structured so that they expand the surface area. So you can see the capillaries on the right-hand side are all spread out, and there are sort of these little lumps that come upward and downward called lamellae. The other thing that helps to make it more efficient is the water is flowing from the front to the back, but the blood is flowing from the back of the gills toward the front of the gills. So the most oxygen-rich water comes in contact with the blood that's highest in oxygen. And then as the water flows across the gills, it gets less and less oxygen in it. So the blood on the back end of the gills has the lowest oxygens, but it can still take up because the oxygen in the water is higher than you see in the blood. So this works in this way. The diagram to the right is a schematic, and it's basically a mirror image. It's flipped around compared to how it is on the left-hand side. But this, we call this a counter-current exchanger because the water and blood are flowing in opposite directions. It's a more efficient way of exchanging oxygen, as opposed to if they flowed in the same direction, you wouldn't notice that much change in the blood oxygen concentration. So for concurrent, it going from a vein to the artery from V to A, you can see that it only increases a little bit, as opposed to the countercurrent going from V to A, it increases a lot because everywhere along that exchanger, it's able to extract oxygen from higher concentration to the lower concentration of the blood. Another thing, we tend to not think of plants as having to take up oxygen, but in marine systems and in marshy systems, the plants that live there tend to have very, very low oxygen concentration in the soils. And so the bald cypress trees in a swamp next to an estuary basically have to have. You may have seen this if you were hiking near First Landing State Park, where there are all these things sticking up out of the ground. Those are called bald cypress knees. If you go out the back end of the Greer building, Around those little ponds, some of the trees that have kind of feathery leaves, those are bald cypress trees. And if you look at the bottom of the soil around them, you can start to see they're starting to grow a few knees there. This helps them transport oxygen down to the roots through this porous tissue called parenchyma. And on the left-hand side, you see it's a cross section of a salt marsh cordgrass. Again, the main plant we see in local salt marshes. This porous tissue helps them transport oxygen down to the roots and helps to aerate it. They need this because while they're photosynthesizing, down in the roots, they have to consume oxygen through respiration and use up some of the calories that they've photosynthesized. So that helps them do that more efficiently. All right, I mentioned the niche before. Habitat and niche are two terms that are often kind of misconstrued or equated with each other. So the habitat is like a physical place where they live or where they feed. Um, so this would be shallow coastal areas versus the niche is the specific role. So the flamingos are going to feed on mollusks from the mud in kind of silty bottom areas, as opposed to dabbling ducks. They're going to feed by tipping, so only shallow plants and only the plants that they can reach without having to submerge their butt underwater. Avocets are going to feed on insects. They have long legs, so they're wading in the shallows, but they kind of move this across the surface back and forth and grab little insects and invertebrates from the surface. Oyster catchers might pry open oysters or mussels with these kind of knife-like beaks that they have. Plovers are going to dart around on the beaches and they kind of pick at little things. So each of them is feeding in the same basic habitat. They have slightly different locations within the habitat that they prefer, and they have a different role. So the niche is more the role of the organism, what it feeds on, where, etc. We might also think of this in terms of what can the organisms tolerate. So this would be steelhead. This is basically it's a rainbow trout that migrates to the ocean. So instead of staying in fresh water all the time, it's born there, the larvae grow up, and then they kind of swim down the river and then they spend time in the ocean. And as a result of this, they can grow much larger than freshwater rainbow trout can, but they then come back to fresh water in order to spawn. So the temperature tolerance, this is a cold water fish, so it really likes it between 13 degrees Celsius and 21 degrees Celsius. Above 21, they're going to be infrequent because they're physiologically stressed, and above 25 Celsius, pretty much average. So 25 is kind of a normal warm water lake. They don't like that. They like cool river water or cold waters. Likewise, if you get down too cold, below 13 degrees Celsius, then that's also stressful. And then basically they're going to be absent below 9 degrees Celsius. So they're going to migrate back and forth, but they're going to stick to this temperature range that they can tolerate. So that's one aspect of the niche. What physical limits do they prefer? Where do they do well? You might think of a warm water fish like a largemouth bass is going to inhabit the same water area when it's in fresh water, but it's going to be on like things on the warmer side, so warm water ponds and lakes and things like that, not cold water rivers. And so that allows there to be more diversity because each of them specializes on a specific area. Now, we might think of this in multiple dimensions. It's not just temperature you have to deal with, but also salinity and maybe the pH range. So this is a hypothetical fish here, and it's showing this in 3 dimensions. This particular one likes 5 to 18 degrees temperature, maybe 6.5 to about 6.8 pH, and then 10 to 20 parts per thousand salinity. So it's going to live in an estuary. It's not going to go in the ocean. It's not going to go in fresh water, but it kind of prefers this temperature range and pH range. Now, what other dimensions are not included? What about the size of its prey? What about how high in the water column? What about the substrate? Does it like sandy areas? Does it like muddy areas? All these different sort of things. Water depth, physical habitat. What's the diet? How big of a food particle is it eating? Large or small? What time is it feeding? In the coral reefs, you have some organisms that feed during the daytime, and then others will rest during the daytime, and then you have the ones that rest during the daytime feed at nighttime. And so they're kind of divided up by time. You have the migrations where they're in different parts of the aquatic habitats during different times. So lots of different things can then separate these different fish and basically reduce competition. So if we think about this in two dimensions, you might have environmental gradient X and environmental gradient Y. And we can show this as either in two dimensions or kind of as the schematic on the right hand side. So for the environmental gradients shown on the left, where that bell curve is highest, that's where they do the best. Just like in that physical tolerance, they like it best in the middle of that range. But think of this in two dimensions, both X and Y. And so if you're comparing these species A and B, A likes there to be less X and less Y. B likes there to be more of both of them. And they do the best within the regions that say high, a little less well in medium, and then low fitness. They're going to have fewer offspring. They're going to have poor survival where it says low fitness. So this can basically reduce niche overlap, and then that helps them reduce competition. So a good example is these two barnacles, Thamylus and Balanus. So Thamylus is the little guy. So if you look on the bottom slide, you can see these tiny little ones and then these big ones that are kind of dwarfing them. The ones that are dwarfing them are Balanus, and then the small ones are called Thamylus. The CH is silent. So anywhere that they live together, and they could potentially live all throughout the intertidal, but anywhere that they live together, the big one, Balanus, is going to squish the little one, Thamylus. So Thamylus is restricted to the higher areas where Balanus cannot exist. Balanus vertically in the intertidal only can go where the blue arrows are, and Thamylus can go where the brown arrows are, but it's restricted to the highest areas because of competition. So fundamentally, it could live throughout the intertidal, but in reality, its realized niche is higher in the intertidal because of competition. All right, now I want to focus on population ecology. So one reproducing collection of individuals that live within a particular location. We're going to use the lionfish because the lionfish is an invasive fish. They're incredibly beautiful in their native habitat, but off the coast of Florida, in Belize, places like that, they are invasive. They're devastating predators. The local predatory fish don't know how to feed on them because they have venomous spines and they don't like getting poked by those. And they eat all the little tiny fish. So a lot of people have gone to try to control the population through spearfishing and things like that to keep them at lower levels in order to preserve some of the other fish because otherwise they just take over the reef. So if you look at them in the blue and in the green, that's where they're supposed to live. Um, uh, Pterois volitans lives in the green area, and Pterois miles lives in the blue area of the Indian Ocean. So that's their native habitat. The organisms there have evolved to be able to deal with them, and they don't get overabundant in any one location. However, with the building of the Suez Canal, some of them have made their way into the Eastern Mediterranean where they are invasive. And because people like them in their aquarium and they want to keep them, but they feel bad when they have to move and they have to get rid of the fish. So they dump it in the ocean, and then this has led to them being invasive all through the Gulf of Mexico, throughout the Caribbean, and up the East Coast, and even down into like around Brazil and things like that. That's the predicted future distribution because there's basically nothing stopping them from moving down the South American coasts to Brazil. So these changes in the abundance of their population, they weren't always this abundant. I've been taking students to Belize since the early 2000s, and when we initially went there, there were no lionfish. But somewhere along the way, somebody released a few of them and they established a foothold. They started breeding and they started increasing in abundance. So the last time we went there, we saw a few lionfish, and I suspect that they are even more now. So these changes in population abundance are basically two factors: biotic potential, which is the capacity of a population to increase, and the things that make the population tend to increase, and environmental resistance, the factors that tend to resist the population. So biotic potential would be each of these guys is going to lay lots of eggs. They have no predators. They have venomous spines to protect themselves. They're well camouflaged. All these tend to increase their survival and increase the survival of their offspring, leading them to increase their population. In contrast, environmental resistance is when something in the environment tends to reduce its abundance. So local groupers learning how to get around the spines and not get stung would be a form of environmental resistance if a predator could actually like deal with them and kind of counteract that. Snorkelers or scuba divers with spearfishing gear. That's a form of environmental resistance, even though it's humans. So how would this change over time? Generally, we think of a population and how it changes over time. It tends to increase sort of exponentially toward the carrying capacity, which is shown here as K. And then sometimes they can overshoot that and then die back. So let's break this down into the two factors. When they're below the carrying capacity, biotic potential tends to want to increase them. So they have the capacity to produce a lot of offspring and reproduce and increase. Once they get over the carrying capacity, there's too many, there's not enough food. This tends to cause them to die off and decrease back down toward the carrying capacity. So I've kind of indicated these on here with the biotic potential in green, environmental resistance in red, just to kind of remind you of that. Now, different populations are going to fluctuate different ways over time. So some populations kind of increase gradually. You see that in the blue line. They have a low intrinsic rate of natural increase, which we denote as little r, and they'll increase kind of gradually until they get to the carrying capacity. If they have a higher capacity to increase, a higher biotic potential, then they're going to overshoot the carrying capacity and then die back. So that's what we call damped oscillation, and that the kind of yellowish neon line, that's showing damped oscillation. Some of them that have very high capacity, so very, very high factor for little r, tends to blip around where the dashed line is, and some of them even more wildly fluctuate over time. But these are natural patterns that we tend to see in living things. So for one that fluctuates a lot, this is phytoplankton. They can double in the span of an hour or hours to days. So they can increase and then decrease pretty dramatically with the response in temperature. Now, before we looked at kind of the seasonal pattern of phytoplankton, even though there's a lot of fluctuation here, you still see this. During the winter months, relatively low concentrations, 4,000 cells per cubic centimeter or less. Then April, the conditions are right. You get the spring phytoplankton bloom, and then there's a spike from 2,000 it bumps up to 6,000, bumps back down, bumps back up because there's a lot of nutrients, but then that causes the zooplankton to grow. And then the zooplankton graze them down, keep them at low levels during May, June, and July. Then the zooplankton start to decline, the phytoplankton increase, and then you get this end of summer peak around 6,000 cells per cubic centimeter, and then they tend to die off into the late fall and into the winter. This follows that what we call a Lotka-Volterra predation model. And generally, the cycles of prey and predators will oscillate or fluctuate over time. There's got to be more prey biomass than predators, so these are on different scales. But generally, the prey will increase first, then that provides food for the predator. So it starts to increase. That increase in the predator then causes the prey to level off and decline. The predator still is increasing because there's lots of food. The prey declines, and then the prey is now at low levels. There's not enough food for the predator, so the predator starts to decline. And it cycles over and over and over again. All right, for community ecology, we're going to get as far as the symbioses on slideshow number 1. We'll save methods of feeding, etc., for the next slideshow. So I want to go through the different types of species interaction: predation, competition, and then symbioses. Symbioses are two organisms that predictably live together for a number of reasons. Okay, so for the species interaction, this can be a consumer resource interaction, a plus minus one. The consumer benefits. This cod is eating a prey fish, and then the prey fish declines. So a predator eats the prey. The consumer is going to increase. Now, for competition, both of them are negatively affected. For mutualism, they both are positively affected. Commensalism, the one species benefits, the other doesn't care. And then for parasitism, one benefits and the other is harmed. So first, we're going to look at the consumer resource interactions. So predator prey. We also have herbivores and plants. Herbivore is where something eats plants as its food. It could be something like kelp, or it could be something like algae, scraping the algae off of a rock, or detritus, a dead decaying organic material. We still tend to think of them in terms of herbivores. A parasite and a host. Parasites often have complicated life cycles that rely on several different ones. So this is a trematode parasite that's going to infect fish. It's going to exit the fish in the feces. The eggs are then eaten by a clam, filtering them out. It then goes through a series of stages. Then it's going to metamorphose into a swimming stage that then infects a brittle star, lives inside the brittle star, and then has kind of a resting stage there that then is going to infect the fish when the fish eats the brittle star. So really long, complicated, usually 2 or 3 different hosts for many parasites. Competition is where 2 species are fighting for resources. We talked about Thamellus and Balanus before. Generally, when they compete, they're both harmed. So 2 plants competing for nutrients, neither of them is going to get as many nutrients as they could by themselves. If 2 fish are competing for food, neither of them is going to get as much food as if they could by themselves. So both species are negatively affected. Mutualism is a type of symbiosis. So this is a predictable interaction where species live together. So mutualism is a plus-plus interaction. The cleaning shrimp is going to benefit, and the moray eel is also going to benefit. The moray eel gets parasites removed by the cleaner, and then the cleaner shrimp gets a free meal. Commensalism is where, another type of mutualism is the clownfish and the anemone. So the clownfish basically is going to benefit from the stinging tentacles of the anemone. It gets protection, and then its waste products help to fertilize the algae that live inside of the sea anemone. Commensalism is where one species benefits and the other doesn't care. So remora have this suction disc and they tend to suck onto anything swimming by. This shark is then has a commensal um, remora attached to the side of it, and the remora is hitching a ride. And when the shark eats a meal, the remora will kind of clean up the crumbs. The shark is not negatively affected by it. Yeah, it's slowed down a tiny little bit, but the remora is really, really streamlined, and so it tends to not be much drag at all. Um, the remora doesn't have to swim, and it gets kind of dragged around to these locations, and it gets to eat the scrap. So it benefits. The shark doesn't care. And then for parasitism, the parasite benefits and the host is negatively harmed. So this is one where there are certain species of isopods that are going to live as parasites within the gills of fish, and they basically ingest the nutrients from the fish and kind of eat away at their gills and stuff like that. And the fish is negatively affected by that.